हेलो फ्रेंड्स। हम स्टार्ट करने जा रहे हैं वन शॉट सीरीज में बायोलॉजिकल क्लासिफिकेशन। सिंस द dawn of civilization, where there have been many attempts to classify living organisms, it was done instinctively and not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more to classify plants into trees, herbs, and shrubs. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups: those which had red blood and those that did not. हम क्या कर रहे हैं, प्लांट्स को मॉर्फोलॉजिकल कैरेक्टर्स के बेसिस पर ट्रीज को, श्रब्स को, हर्ब्स को डिवाइड कर रहे हैं अलग अलग कैटेगरीज के बेसिस पर। डिवाइडेड एनिमल्स इनटू टू ग्रुप्स, जो दो एनिमल्स को हम दो ग्रुप्स में डिवाइड कर रहे हैं, एक तो रेड ब्लड और जिनका रेड ब्लड नहीं है उनका। इन linear time or two kingdom system of classification with plenty and animal was developed that included all the eukaryotes and prokaryotes, unicellular and multicellular organisms. Unicellular and multicellular animal organisms and photosynthetic green and non-photosynthetic fungi done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat. Systems for the living organisms have hence undergone several changes over the time through plant and animal kingdoms have been constant under all different systems. The understanding of what groups organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. Monera, Protista, Fungi, Plantae, and Animalia, 5 kingdom. Characteristics of 5 kingdom classification. Cell type: Monera, prokaryotes. Cell type: Protista, eukaryotes. Fungi, eukaryotes. Plantae, eukaryotes. Animalia, eukaryotic. Cell wall: Monera, non-cellulosic polysaccharides plus amino acid. Protista, present in some fungi, present with chitin. Plantae, present cellulose. Animalia, absent. Nuclear membrane, absent. Monera, absent. Protista, nuclear membrane present. Fungi, nuclear membrane present. Plantae, nuclear membrane present. Animalia, nuclear membrane present. Body organization: Monera, cellular. Protista, cellular. Fungi, multicellular. Plantae, tissue organs. The animalia, tissue organ systems. Mode of nutrition: autotrophic in Monera. Protista, autotrophic. Fungi, heterotrophic. Plantae, autotrophic. In Animalia, heterotrophic, holozoic, saprophytic, etc. R.H. Whitaker proposed a five kingdom classification. The kingdom defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three domain the system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain, and thereby a sixth kingdom classification. You will learn about this system in detail at higher classes. Let us look at this five kingdom classification to understand the issues and construction and considerations that are influenced the classification systems. Earlier classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character, that they unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Any, for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic under algae. The classification did not differentiate between the heterotrophic groups fungi and the autotrophic green plants. Through they also showed characteristics difference in their walls composition. The fungi had chitin in their walls, while the green plants had it cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista had brought together Chlamydomonas, chlorella, earlier placed in algae within plants, and both having cell walls with paramecium and amoeba, which were earlier placed in the animal kingdom, which lacks cell wall. It has put together organisms happened because the criteria for classification changed. This kind of changes will take place in future to depending on the improvement in our understanding of characteristics and evolutionary relationships over time. An attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities, but is also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whitaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt separately in chapter 3 and 4. Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans. where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical Coccus cocci, rod-shaped Bacillus bacillia, and comma-shaped Vibrio, and spiral Spirillum spirilla, which is located in figure 2.1. फर्स्ट है हमारा कॉक्सी, जो कि है स्पाइरल शेप्ड कॉक्सी, जो कि है हमारा कॉकस शेप्ड बैसिलिया, जो कि है हमारा बैसिलस रॉड शेप्ड स्पाइरिला स्पाइरल शेप्ड विब्रियो कॉमा शेप्ड. Though the bacterial structure is very simple, they are very complex in behavior compared to many other organisms. Bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic, autotrophic, or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats, such as extreme salty areas, halophiles, hot springs, thermoacidophiles, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having the different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane from the dung of these animals. Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A, similar to green plants, and are photosynthetic autotrophs, which is located in figure 2.2, a filamentous blue-green algae nostoc. The cyanobacteria are unicellular, colonial, or filamentous, in the freshwater, marine, or terrestrial algae. The colonies often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts, for example, nostoc and anabena. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia, and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legumes, roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Chlorella, typhoid, tetanus, citrus, canker are well known diseases caused by different bacteria. Bacteria produce Bacteria reproduce mainly by fission, figure 2.3 sometimes, dividing a bacterium. Under unfavorable conditions, they produce spores. They also reproduce by the sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. And the mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in nature in animals and plants. Kingdom Protista. All single celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protista on to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime moths, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the other dealing with plants, animals, and fungi. Being eukaryotes, the protostan cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protistas reproduce asexually and sexually by the process involving cell fusion and zygote formation. Chrysophytes. This group includes diatoms and golden algae desmides. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water currents, plankton. Most of them are photosynthetic in diatoms. The cell walls form two thin overlapping cells which fit together as in a soap box. The walls are embedded with cilia, and thus the walls are indestructible. And thus, the atoms have left behind a large amount of cell wall deposits in their habitats. This accumulation over billions of years is referred to as diatomaceous earths. Being gritty, this soil is used in polishing nitration of oils and syrups. Diatoms are the chief procedures in the oceans. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, and brown, blue, or red depending on the main pigment present in their cells. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the say wall plates. Very often, red dinoflagellates, for example, Goniolex, undergo such rapid multiplication that they make the undergo such rapid multiplication that they make the sea appear red tides. Toxins released by such large numbers may even kill other marine animals such as fishes. Euglenoids, majority of them are freshwater organisms found in stagnant water instead of a cell wall. They have a protein rich layer called a pellicle, which makes their body flexible. They have two flagella, a short and a long one. Through they are photosynthetic in the presence of sunlight. When deprived in sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants, for example, Euglena, which is located in figure 2.4b. Euglena. Slime modes. Slime modes are saprophytic protistas. The body moves along decaying twigs and leaves. Euglena engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several facts. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. The spores possess two walls. They are extremely resistant and survive for many years. Even under adverse conditions. The spores are dispersed by air currents. Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to the primitive relatives or animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia, a false fact as in amoeba. Marine forms have silica shells on their surface, some of them, such as antamoeba or parasites, flagellated protozoans. And the members of this group are either free living or parasitic. They have flagella. The parasitic forms cause diseases such as sleeping sickness, for example, trypanosoma. Ciliated protozoans, these are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be stirred into the gullet. For example, paramecium, which is located in figure 2.4d. Sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium malarial parasite, which causes malaria, the disease which has a staggering effect on human population. Kingdom fungi. The fungi constitute the unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and and toadstools are also fungi. White spots seen on mustard leaves are due to parasitic fungus. Some unicellular fungi, that is, for example, yeast, are used to make bread and beer. For other fungi cause diseases in plants and animals. Wheat rust causing Puccinia is an important, for example. Some are the source of antibiotics, for example, penicillium fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid. It is to prevent food from going bad due to bacterial or fungal infections, with the exception of yeasts, which are unicellular. Fungi are filamentous. Their bodies consists of long, slender, thread-like structures are continuous tubes filled with multinucleated cytoplasm. These are called conocytic hyphae. Others have septae and cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depend on living plants and animals are called parasites. They can also live as on living plants and animals are called parasites. They can also live as symbionts in association with algae as lichens and with roots of higher plants as mycorrhiza. Reproduction in fungi may take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spores called conidia and sporangiosperms or zoospores, and sexual reproduction is by oospores, ascospores, and basidiospores. The various spores are produced in distinct structures called floating bodies. The sexual cycle involves the following three steps: fusion of protoplasms between two motile and non-motile gametes called plasmogamy, fusion of two nuclei called karyogamy, meiosis in zygote resulting in haploid spores. When the fungus reproduces sexually, two haploid hyphae of compatible mating types will come together and fuse. In some fungi, the fusion of two haploid cells immediately resulted in diploid cells. However, in other fungi, ascomycetes and basidiomycetes, an intervening this dikaryotic stage in n plus n, that is two nuclei per cell, occurs. Such a condition is called dikaryon, and the phase is called the dikaryophase of fungus. Later, the parental nuclei fuse and the cells become diploid. The fungi form fruiting bodies in which reproduction division occurs, leading to formation of haploid spores. The morphology of the mycelium, mode of spores formation, and fruiting bodies form the basis for the division of the making of the kingdom into various classes. Phycomycetes. Members of Phycomycetes are Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. The mycelium is aseptate and conocytic. Asexual reproduction takes place by zoospores motile or by aplanospores non-motile. These spores are endogenously produced in sporangium. Zoospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous, or example of are dissimilar anisogamous or oogamous. Some common examples of mucor, which is located in figure 2.5a. Rhizopus, the bread mold mentioned earlier, and albugo, the parasitic fungi or mustard. Ascomycetes, commonly known as sac fungi. The ascomycetes are mostly multicellular, for example, penicillium, or rarely unicellular, for example, yeast, Saccharomyces. They are saprophytic decomposers, parasitic or coprophilous, growing on dung. Mycelium is branched or septate. The sexual spores are conidia produced exogenously on the special mycelium called conidiospores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like acts. Singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are aspergillus, figure 2.5b. Aspergillus, claviceps, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members, like morels and truffles, are edible, are considered delicacies. Basidiomycetes, commonly known forms of basidiomycetes, are mushrooms, bracket fungi, and puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as parasites, for example, rust and smolts. The mycelium is branched and septate. The sexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about the fusion of two gamma vegetative or somatic cells of different strands of genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis takes place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus mushroom, figure which is located in 5.2.5c, Astrelago, smut, and Puccinia rust angus. mushroom. Deuteromycetes, commonly known as imperfect fungi, because only the sexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they are rightly belong to. It is also possible that the sexual and vegetative stage have been given one name and placed under deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of deuteromycetes. Once perfect, the sexual stages of members of deuteromycetes were discovered, they were often moved to ascomycetes and basidiomycetes. The deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is separate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers or litter and help in mineral recycling. Some examples are Alternaria, Coletotrichum, and Trichoderma. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members of are partially heterotrophic, such as the insectivorous plants and parasites. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cuscuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplasts of cell wall, mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycle of plants have two distinct phases, the diploid saprophytic and the haploid gametophytic, and that alternate with each other. The lengths of the haploid and diploid phases and whether these phases are living free living and dependent on others vary among the different groups in plants. This phenomena is called alternation of generation. You will study further detail in this kingdom of chapter, which is chapter 3. Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular, and their cells lack cell walls. They directly or indirectly depend on plants and for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition to holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite steps, shape, and size. Higher forms slow higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The asexual reproduction is by copulation, of male and female, followed by embryological development. Salient features of various phyla are described in chapter 4. Viruses, viroids, prions, and lichens. In the five kingdom classification of Whitaker, there is no mention of lichens and some cellular organisms like viruses, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu known what effects viruses can have on us, even if we do not associate it with our condition. Viruses did not find a place in classification since they are not considered very truly divine living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Tobacco mosaic virus (TMV) bacteriophage. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing killing the host. You would you call the viruses living or non-living? Virus means venom or poisonous fluid. Dmitry Ivanovsky in 1892 recognized certain microbes as causal organisms of the mosaic disease of tobacco, which is located in figure 2.6a, tobacco mosaic virus (TMV). These were found to be smaller than bacteria because they passed through bacteria proof filters. M.W. Beijerinck in 1898 demonstrated that the exact extract of the infected plants of tobacco could cause infection in healthy plants and named the new pathogen virus and called a fluid as contagium vivium fluidium infectious living fluid. The W.M. Stanley in 1935 showed that viruses could be crystallized, and crystals consist large of proteins and they are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single structured RNA, and viruses that infect animals have either single or double structured RNA or double stranded DNA. Bacterial viruses or bacteriophages, viruses that infect the bacteria, are called capsid, made of small subunits called capsomers. Projects the nucleic acid. These capsomers are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by the virus. In plants, The symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, Theo Dinier discovered a new infectious agent that was smaller than the viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked it lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was also of low molecular weight prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded proteins. The agent was similar in size to viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle, and it is analogous variant CR Jacob disease CJD in humans. Lichens are symbiotic associations, that is mutually useful associations between algae and fungi. The algal components is known as phycobiont and fungal components is called as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi, and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw an lichen in nature, one should never imagine that they had two different organisms within them. Lichens are very good pollution indicators. They do not grow in polluted areas. Summary. सो लास्ट में हम समरी देखेंगे बायोलॉजिकल क्लासिफिकेशन के. बायोलॉजिकल क्लासिफिकेशन ऑफ प्लांट्स एंड एनिमल्स वाज फर्स्ट proposed by Aristotle on the basis of similar morphological characters. Linnaeus later classified all living organisms into two kingdoms, plant and animalia. Whitaker proposed an elaborate five kingdom classification: Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of the five kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationships. In the five kingdom classification, bacteria are included in kingdom Monera. Bacteria are composite cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic and heterotrophic in their mode of nutrition. Bacteria kingdom Protista includes all single-celled eukaryotes such as chrysophytes, dinoflagellates, euglenoids, slime moths, and protozoans. Protistas, both sexual and sexual members of kingdom fungi, show the great diversity in structures and habitats. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, ascomycetes, basidiomycetes, and deuteromycetes are the four classes under this kingdom. Under this kingdom, bryophytes, pteridophytes, gymnosperms, and angiosperms are included in this and saprophytic generations. The heterotrophic eukaryotic multicellular nutrition of these organisms are holozoic. They can reproduce mostly by the sexual mode. Some say cellular organisms like viruses and viroids, as well as the lichens, are not included in the five kingdom system of classification. So यहाँ पर हमारी समरी ख़त्म होती है बायोलॉजिकल क्लासिफिकेशन की, जिसमें हमने देखा फर्स्टली किंगडम, फाइव किंगडम क्लासिफिकेशन देखा जो आर.एच. विटाकर ने दिया था। उसके बाद किंगडम मोनेरा, आर्कीबैक्टीरिया, यूबैक्टीरिया, किंगडम प्रोटिस्टा, क्राइसोफाइट्स, डायनोफ्लैजलेट्स, यूग्लेनॉइड्स, स्लाइम मोड्स, प्रोटोजोन्स, किंगडम फंगाई, फाइकोमाइसेट्स, अस्कोमाइसेट्स, बेसिडियोमाइसेट्स, ड्यूटेरोमाइसेट्स। उसके बाद हमने देखा किंगडम प्लांटी, किंगडम एनिमिलिया, वायरसेस, विरोइड्स, प्लांट्स एंड लाइकन्स। उसके बाद हमने TMV टोबैको मोज़ेक वायरस देखा। उसके बाद बैक्टीरियोफेज और उसके बाद समरी देखी। तो यहाँ पर हमारा बायोलॉजिकल क्लासिफिकेशन चैप्टर होता है ख़त्म। सो थैंक यू फॉर वॉचिंग।